Howdy, AP Bio. Welcome to the real Day 65. You can see that you would have been doing GR 44 and part of 43 to get ready for today. At the end of this, you'll go after the rest of 43 in the immune system, so you prepare for what we're doing next week. We would have done a Goldfish Capillary Lab, which is really fun, and too bad we can't do it this year, but you can always try it at home. And then, of course, the 2013 and 2016, we'd be dealing with those. The next review session is on the 3rd, right? So one thing that we can see as our goal today would be to understand how things like drugs, like stimulants and depressants affect the cardiovascular system, to get an idea how the cardiovascular system works. And then importantly, how you actually stabilize your pH, because it's a fascinating story and involves chemistry. And, that's what the AP loves and doctors love too. So if we look at this, always remember to make sure that you live your life with purpose. My purpose right now is to give you videos that help you to understand this stuff and to annoy you with all my bad jokes. So I'm going to get rid of my ugly face. Boom. And what we're going to dive into, of course, is some notes first. And the first thing we should recognize is there's some confusion about NAD plus versus NADH. So you should sketch this down into your notes to help yourself out. If we look at NAD plus, it, of course, is the electron stripper. It strips 2 electrons and 1 proton, a hydrogen ion, from anything, of course, that it can get its hands on and actually rip that thing off because it's an oxidizer. And then NADH is the electron carrier. It also carries 1 proton. So ways to think about these is we call the NAD plus a stripper because it strips electrons away from high energy molecules. But that's not what you should call it in an exam. You should call it an oxidizer or an oxidizing agent. It steals electrons. It oxidizes things. NADH, of course, is a giver. It's a reducer. It gives electrons to other molecules such as the ETC. And that means that it gives energy to cause changes, to make life happen. So things that we should recognize, this is how you remember it. It's a stripper and a giver. This is what you call it in an AP, an oxidizer or oxidizing agent, right? And a reducer, a giver of electrons versus a stealer of electrons. This all goes along with a handy thing from chemistry called Leo says Ger. And if we look at Leo says Ger, it's written funny because if you lose electrons, that's oxidation, Leo. And then we look at this, this is like glucose. Glucose is oxidized by NAD+, and that means that NAD+, the stripper, is reduced. It gains the electrons because it's doing the oxidizing. It's the oxidizing agent. So it kind of makes you go cross-eyed. Remember, the oxidizers like oxygen. You can always think about oxygen being the electron hog. It's going to steal electrons. Things that oxidize act like oxygen. If we look at the Ger part, that's if you gain electrons, that's reduction. So if I'm looking at the ETC, the electron transport chain is reduced by NADH because it's giving its high energy electrons, which oxidizes NADH. So the ETC oxidizes the electron carrier because everybody's always going to be taken when the other one's given. So if we look at this, NADH is my reducing agent, which allows, of course, it to give electrons to the ETC so it can pump those protons. Boy. No wonder this is confusing to some people. But just remember, oxidizers take electrons, reducers give them. And don't forget, with photosynthesis, it's NADP plus NADPH when we're looking at what's going on in photosynthesis. So you can always think about reducing agents like Neil. He's that loser that gives you energy and ideas, hopefully, right? Okay. Now, key thing is we're going to dig into our ETD now, so you might want to flip to a new page. This, as you hopefully know by now, is my oldest son, Finny. And good old Finn was actually named after his ancestor, the fish, but don't tell my wife. And this little goldfish here has fins. And in our lab today, we would be looking at the fins of this fish because inside of them, we can see the capillaries that we evolved our capillaries. That's why his face is partly blushed here. Those little capillaries run to every single cell tissue in our body that's good and healthy, except for some of the joints. And we evolved our traits from the fish. So today we'll be working with the fish to understand how capillaries can change how our cardiovascular system works. To get that, we would notice that their blood is red because their blood also contains an amazing protein called hemoglobin. What you're going to do for your ETD, and I'm going to help you with, is you're going to draw and explain the roles of hemoglobin, bicarbonate ions, HCO3 1-, and this stuff called protons in gas exchange. To use it as a template, you should draw this picture. Hopefully you can recognize some parts of it, right? Nice and big on a whole page of your ETD because we're going to fill this bad boy in. Right? And then you're going to attempt to draw and explain in steps what the role of each of those things does in this. So pause me, try it out on your own, And now we're going to do it together. So if we look at this, one thing we can notice is we got to get a lay of the land of what's going on here. And what this is right here, this is your alveolar space. So this is those little tiny air pockets that are inside your lungs, which give you tons of surface area. That's contact with the outside atmosphere. At the same time, we can see we've got this thing. This, of course, is a red blood cell, an erythrocyte. And that red blood cell is carrying an amazing molecule called hemoglobin. And that's what this is. This is hemoglobin. Now, hemoglobin is a functional protein, and it does many, many things. And the most famous thing most people know is it carries oxygen in your blood. It has 4 active sites that are actively involved in this. So there's more going on. So it's pretty much always carrying some oxygen because inside of this thing is a heme group. And there are 4 heme groups, and heme is referring to hematite, as in iron. That's the part of it that, of course, rusts and grabs onto oxygen. That's why places like Australia and Africa are rusty red, because they have iron in their soil. So it becomes iron oxide. You literally rust your blood. That's why it's red. Now, key thing with this, of course, is this blood is going around inside of your lungs right now. So it's in the capillaries of the alveolar space. And that's great because you just took in a breath, which means you just filled the alveolar space with lots and lots of oxygen. This is a high oxygen concentration. And if things are going really well, that means that this is a low CO2 concentration, right? There's not a lot of CO2 because you're taking a fresh breath, unless you're sitting in a closed garage, but don't do that. Now, key thing of this, of course, stuff will always push from where there's more of it, a high O2, to your blood, which has a relatively low O2. So it's going to push from high to low down its partial pressure gradient. Each gas has its own pressure gradient, but it always goes from high to low, right? There is no suck in science. There is only push. As it does that, the hemoglobin has a free active site with a heme. At least one of them is going to be open at this phase, and it will grab onto that oxygen, and now that blood is fully loaded, though there's lots of hemoglobin molecules all over it. At that point, it's so rusted that your blood would be bright red. And that's why if you're a hunter, you know when you get a lung shot, because you get really bright red blood, and the pH of it, crucially, is exactly 7. Now, that 7 is because, of course, it's neutral. That's good. That's what a lot of your proteins in your body work best at, neutral like water, right? Now, that's loaded up with oxygen, but you got to take this and get this to the rest of your body. So now this is going to go to your heart. It's going to travel through the pulmonary vein, back to the heart. It's made its way into the left ventricle, and then it gets pumped out from the heart to the system, right? It's going to go to the systemic circuit. Now, the key thing is we're coming from the heart, and we're going to the systemic circuit so we can feed things like your brain, your liver, your digestive organs, your muscles and stuff. And it's the same blood cell because it's going in a circulatory system, and it's still got the same hemoglobin, right? With its active sites, it's loaded with all those oxygens, and it's ready to do its job, right? This would be stage number 2. This is stage number 1, but it's a cycle, so whatever. This, of course, is not in the lung. This is in your tissues. These are tissue cells. And being tissue cells, of course, they're the ones doing the work for your body. They are bathed in this stuff called interstitial fluid because you're mostly water, and your cells, of course, are attached to each other, but there's fluid in between them, so they're constantly connected by a solution, right? Now, these cells are doing something crucial. They're living, so they need energy. They're doing cellular respiration. Since they're doing cellular respiration, they're taking sugar and they're burning it, and they're producing a lot of CO2. It's in the interstitial fluid. It's inside the cell's cytoplasm itself. This is a high CO2 concentration because they're working. They're making smoke. At the same time, they're burning their fuel, so they're running low on oxygen. That's why you take a breath, right? Now, the cool thing about this is that means that there's high oxygen inside of your blood cells, and there's low inside your tissue cells, so it's just going to push from high to low and give the cells what they need with no energy output or input at all, because it's just pushing down its gradient as long as you got nice, big, fresh breaths. Now, when that does that, of course, there's a cool thing that happens. That red blood cell has now dumped off part of its cargo. It's still got the same hemoglobin. It's still loaded with most of the oxygens, but the one active site has opened up. And that's crucial because hemoglobin doesn't just carry oxygen, it carries lots of other things, right? Now, we can see that there's 3 different things that are going on here. There is what is called the blood plasma. That's the fluid that's running around, a series of proteins and a whole bunch of water, right? And then there's the actual cytoplasm of the cell itself, and there's the hemoglobin too. So the neat thing is there's a bunch of CO2 building up outside in the tissue cells and not a lot in the blood, so it's going to push from high to low. And when it does that, it goes into a couple places. One place it goes is some of it goes directly into the blood plasma. That's why, of course, you can take plasma transfusions, and it's still good for your body, right? The other place that it's going to go is it's going to go directly into that open active site on the hemoglobin because it's got oxygen in it. So it can go after anything that's electronegative, and it grabs onto that thing, which is nice. At the same time, some of that CO2 will go directly into the cytoplasm of the blood cell itself. And without a nucleus, there's a lot of goo in there. And most of that goo is water. Now, the neat thing is you're basically making a soda. The CO2 will come in and it will collide, because this is chemistry, with the water itself. And it will go into solution. And just like in chemistry class, it will become carbonic acid. It's the same thing that leads to ocean acidification. Now, this being carbonic acid, what it's going to do is what all acids do. It's going to do, of course, proton football. It's going to dissociate, and when it dissociates, it's going to create a bicarbonate ion, and it's going to throw out the football, which is the protons. Now, crucially, when that happens, that means that we've got protons in solution. That's why it's the pouvoir de hydrogen, the power of hydrogen, that would give us the power of the acid. This would cause your blood's pH to actually decrease because it's becoming acidic. Now, you can tell this if, of course, you hold your breath, you would start to feel your body change in chemistry as you start to feel that burn and you desire breath. That's your hypothalamus being told to breathe for you, right? This means that our pH will now drop below 7 and the blood will become darker red because it's not as oxygen rich. It's not as rusted. This is when if you see a gunshot come into the ER and it's dark blood, then you got a pretty good sign that it didn't hit the lung. That's blood that's making its way back to the lungs because it's lost its load of oxygen, right? Now, the heart's going to keep beating. And it's spent lots of time in the capillaries because there's a lot of surface area, which gives it time for all this diffusion. But the blood cell's still doing some magic. This would be step number 3. Here's step number 4. Now, there's a problem with your pH dropping. If your pH in your blood drops too far, that can denature the proteins throughout your body, which basically run all of the cell membranes and your brain. And that means that, of course, it could kill you because your cells wouldn't be functional anymore. So that's when hemoglobin steps in and does another magic trick. Now, the plasma is still carrying CO2. And the other stuff that went into the cytoplasm, that had the bicarbonate. The bicarbonate actually diffuses out of the cell. At the same time, the hemoglobin, by picking up that CO2, because it changed conformation again, as soon as CO2 goes into it, it changes shape. And that opens up another active site that actually reaches out and grabs on and tracks over the protons, taking the protons out of solution, which makes your pH go up a little bit. Now, at the same time, it took the other part of the carbonic acid, this bicarbonate thing, and it put it into the solution in the blood plasma. Now, the awesome thing about that is bicarbonate is famous. You've heard of sodium bicarbonate. Hey, that's baking soda. That's a base. But what it really is it's a buffer. It's a weak base, which can function as an acid if it needs to. It can play proton football. But here in this situation, we have too many protons. So the cool thing is the hemoglobin does a great job. It helps to keep the actual blood pH from getting too low. At the same time, this bicarbonate acts as a buffer and will grab up any extra protons, which makes the pH go up a little bit so that it doesn't get too acidic. Now, it's still going to be a little bit acidic, but not as acidic as it would be because you're buffering it with bicarbonate and the hemoglobin's grabbing the protons at the same time. You still got CO2 into your blood plasma, so you're still going to have some acid, but not as acidic as it would be, saving your life. Now, this blood is loaded with CO2 as well as the plasma because it's doing work for you too. It's going to make its way back from the tissues, shooting through the heart again. And as it makes its way to the heart, it then, of course, makes its way back to the lungs. When it gets back to the lungs, some amazing chemistry happens. One, we can see that it's still got the hemoglobin. The hemoglobin, as always, is still carrying some oxygen because it doesn't like to let go of iron. It has CO2 that is carrying, and it has protons that is carrying too. The plasma is carrying the CO2 as well as it's carrying other CO2 in a safer form as a buffer that saves your life, right? Now, the crazy thing is when it gets back to the lung, if you've taken a fresh breath, this is now a high CO2 concentration inside of your blood, and there's a low outside. So CO2 wants to go from high to low, and it pushes down its partial pressure gradient. That means that the CO2 in the plasma will leave on its own, no energy expended as it moves towards the low. Hey, getting rid of some of that stuff, right? At the same time, a really cool thing happens. As the partial pressure changes, what happens is the hemoglobin, it senses that pressure change. And that decreases its affinity, its attraction for the CO2 that it's carrying. And what it does is it actually lets go of the CO2, which then can move out into the solution and leave the cell. Now, the great thing about that is when it happens, that's all due to the partial pressure change. That's the beauty of hemoglobin. It's a little octopus that can change its shape and change its function. It's a functional protein with many functions. That's why the AP loves it, and your body loves it too. Now, the neat thing is, it also, of course, changes conformation when it loses the CO2, and it spits out the proton it was holding on to before, which goes back into the plasma, which means it can then collide with the bicarbonate again. And when it does that, it becomes carbonic acid. Because of Le Chatelier's principle, if you have these and you add them, they're going to run away and they become the carbonic acid. At the same time, there's CO2 leaving the body. So that makes, of course, Le Chatelier's principle kick in again, where if you don't have enough CO2, if you take it, it moves towards and it produces the CO2 and the water. That CO2 comes out of carbonic acid, bubbles out of your blood into your lung space, leaves your body, and leaves you with H2O and an open active site. So you're back at station number 1 with lots of water, lots of plasma, and now your pH is back to magical 7, right? Notice that this partial pressure change is pretty important. Your protein can sense the pressure change plus the number of collisions that you have. Now, the cool thing is I've made on here an animated version of it on the notes for the PowerPoint as well. So you can follow through those steps again, and you can actually read it. You should make sure that you check that out because it's pretty handy and pretty darn complex. It's probably the most complex thing that you have to learn this year, besides, say, the immune system and the endocrine system. In addition to that, of course, you can freeze frame it with that. So check out the PowerPoint if you want to be able to read it better. If you look into GR42, which we're not done yet, if you hopefully noticed, kind of like cardiovascular system is important for Americans. If you flip to page 10, it really starts talking about the circulatory and the respiratory system, right? One thing we can notice is that we use lungs to breathe. We have these wonderful things called alveoli. They are loaded with capillaries to increase surface area. More surface area, more time, and more chance for diffusion. It's got to stay wet. If you fill it up with a bunch of mucus from coronavirus, hey, guess what? You don't get enough oxygen and your organs start to fail. Now, this has anything to do with real life. Now, key things we can see is to talk about this partial pressure. There's more oxygen if you take a big fresh breath outside, so it pushes in. There's less CO2 outside if you've been working hard, and it pushes out. If you put a bag over your head, of course, then the CO2 is going to build up and it would suffocate. So don't do that, right? It's all about them moving down their partial pressure gradients. They always go from high to low, right? This is the image that's inside the book that we just described. It shows the story, but it shows it a bit more complex, so hopefully you understand a little better idea of how it's working. One thing we can see with the fish is they're using their gills, but they're also ramming themselves forward. And the neat thing is, as they use their gills and ram ventilation to let fresh water with oxygen-rich water flow over their gills, they push their blood in the opposite direction so that it pushes the CO2 out in one direction and forces the oxygen in another. It's that counter-current exchange that aids the partial pressure gradient. And that's why they got to keep moving those fishy-faced gills, and they're a operculum, right? Now, what are capillaries, and why is it important for them to be small and thin-walled? Capillaries, hopefully you realize, are the smallest blood vessels in your body. They come in contact with pretty much every cell in your body except for some of your joints. They have to be small and thin because that allows diffusion to happen. Now, if we look at one of the best images they tend to show in an AP, or at least in medical school when you run into it, there's this diagram where we're going from arteries, arterioles, to capillaries, to venules, to veins, right? As it's coming in, there's some neat things that happen. You can see those tiny little capillaries have tons of surface area. Hey, that's more area for gases to diffuse and nutrients to diffuse, like glucose. That increases the ability to do diffusion. Surface area is all about it, right? More collisions. The velocity of the blood, of course, is really high in the arteries because it's coming straight from the heart, away from the heart. Hits the capillaries, they're tiny tubes. Things slow down. It's like being stuck in the lunch line, right? But that's good because that aids diffusion. It takes time. Think back to the old Dbag and Tater Hater labs. It took a whole set of days for the diffusion to be apparent. As it comes back, of course, it speeds up a bit in the veins because they're larger, but that's because you're moving around your legs to help pump it back to the heart. But you lose a lot of that velocity, and that goes along with the pressure. You can see your systolic, there's your heart, there's your back pressure, the diastolic, and then you can see as we get into the capillaries, pressure drops dramatically, and that's a good thing because you want some pressure, but you don't want to below these little thin capillaries. And then it really drops when you get back to veins because you've lost the push of the heart. And that's why veins have to have valves, and your skeletal muscles help to force them back. So this diagram, they love to ask questions about it and say, hey, what's going on with the blood vessels? Why does it look this way? Hopefully you understand it's about slowing things down, making sure you get a chance to have diffusion. Now, you can also change what your capillaries are doing because you have these control valves called the PreCapillary sphincters. And those sphincters can open up or shut off the blood flow inside the capillaries. At the same time, you have smooth muscle in the walls of those and your arteries that can contract or relax and control the blood flow of that. The blood will always keep flowing through main venules and arterioles, but the capillaries can shut off and open up. And you can see that anytime somebody blushes, so their capillaries are being flushed. That would be vasodilation versus them being pale. That's vasoconstriction. Now, this can happen if you're ever walking in the woods like I was up in Banff and you run into a grizzly bear. Now, when you see a grizzly bear, of course, what does your body do? Well, it does a couple things. One thing is your heart races faster, your blood pressure increases, your pupils actually dilate, your muscles can actually move faster because they have more blood flow, your brain can think faster, and people pee their pants. At the same time, their skin goes pale because there's a lot of changes happening to their circulatory system. Sometimes they even poop their pants. When I saw the bear, all I did was say, hey, bear, and then I took some pictures, right? Now, key thing is that's because this is kicking off what's called your fight or flight mechanism, what's known as the sympathetic nervous system. Your spinal cord is basically changing a bunch of activity of your circulatory system and your respiratory system. It's increasing and dilating your bronchi so you can get more oxygen to your tissue so you can run and think faster. That allows your brain to think faster. You can actually operate, and it seems like time slows down because you're thinking faster. Your pupils dilate so you can see better, especially at nighttime when most of our predators would have been in the past. Your heart races to increase blood flow to those tissues. You actually start breaking down fat so that you can supply yourself with energy. You contract things like your bladder because that would, one, decrease the weight of your body, allowing you to run faster, and actually make you not taste as good. Skeletal muscles become stronger and firmer. In fact, pain receptors are shut down at this time. That's why mommies that have a car that backs over their little kid can pick up the kid, tear out their muscles, and not even feel it. They're in shock. What they're really in is they're kicked in their sympathetic nervous system. And your GI tract will shut down. You don't do digestion. In fact, you expel some of it so you can drop your weight as well. So as well as your liver's kicking in a whole bunch of glycogen to turn it into glucose, and you start sweating so you don't overheat as you're running away. All those changes happen really, really fast because they were controlled by your sympathetic nervous system. Now, those are the noticeable effects, but there's also a bunch of hormone and blood pressure changes that you don't notice so much that are controlling these things. We'll get more into hormones later. The reason it happens so fast is it skips your brain. It's controlled by your spinal cord, which is the most primitive form of your central nervous system that we inherited from those fish. It's a good way to continue to survive. There's also a thing called the parasympathetic nervous system that works in parallel to it, beside it, and can counter some of these and do some other things. But the sympathetic is all about fight or flight, survival. And it really is about controlling where the blood flows, right? So that is back to the story of the old elephant seal. He was able to dive deep because he could change his blood flow. He's not thinking about it actively. His spinal cord is going into the reaction of shutting off blood vessels in the extremities, increasing the amount of blood flow to his muscles and to his brain so that he can think, see, and move underwater at great depth and not die from oxygen depletion. Kind of cool what it can do for you. Now, today what we'll be doing in lab, and it's a really fun lab, is we'll be looking at goldfish. Now, if you let goldfish in the wild, they are Asian carp, and they can grow to massive sizes. Our little goldfish, of course, we've been doing some stuff with. One of the things we'd be doing with them is be giving them drugs. And when we talk about drugs, we'd be giving them two drugs, nicotine, a stimulant, and alcohol, a depressant. And we wouldn't be feeding it into their faces, we'd be putting it onto their tail. And by doing that, we could do a really cool thing. We'd basically build fish tacos and then drop little drops of the two different stimulant and then the alcohol onto their blood vessels that are inside their tail. And the neat thing is when you do that, you can actually see under the microscope their blood cells moving through. And I'll show you this quick video so that you can get an idea of what it would have looked like if you could see it. This was made by Stevie Taylor. He was a 7th grader, and some seniors have trouble doing this stuff. Come on, baby. And now it's going to freeze. Maybe it doesn't like YouTube when you do screen casting. I don't know. Okay, whatever. You can check out that. The links for it are on this. Worked when I tested it out, but not this time. There's the best link for it to show you what it looks, and you can see the actual blood vessels open up, the capillaries. That would be vasodilation. Or you can see them shrink down in this video, which is vasoconstriction. Now, key thing is when we look at the fish, you would have added two drugs. One of them would have vasodilated and opened up and increased blood flow. The other would have vasoconstrict and decreased blood flow. But you can see it in the videos you're watching. Now, which one, alcohol or nicotine, would cause it to have these effects? Well, the vasodilation means more blood is going to the periphery. So that is a depressant. That's the alcohol would make it vasodilate. Vasoconstriction would reduce the amount of flow to the extremities. That's what the nicotine, the stimulant, does. And this all goes back to the idea of norepinephrine and the fight or flight thing. Our sympathetic nervous system runs on norepinephrine, which is an isomer of this lovely stuff called epinephrine, or you've heard of it as adrenaline. It's the same stuff we find in EpiPen, an adrenaline shot, when somebody has a problem with their lungs not functioning correctly, or they get stung by a bee and they go into anaphylactic shock. Basically, what these stimulates, like nicotine and caffeine do, is they stimulate your body to release its own norepinephrine. And what that kicks off is that's an adrenaline rush to go into fight or flight. And what happens is you will vasoconstrict the blood vessels on the extremities and go pale. And that means that the blood then will rush to your brain and to your muscles so that you feel like you're more alive, you increased your energy. But really what you're doing is you're burning off your sugar reserves much, much faster. And that's why you get the caffeine crash if you try that. That's also why people that are smokers tend to have really, if they've been smoking a long time, really pale, gross-looking skin and pale, gross-looking hands because they've been vasoconstricting the outer extremities of their body for so long that some of them are starting to die away and be depleted of nutrients. At the same time, of course, they do it because they're addicted to that rush of adrenaline that makes their brain feel awake. But that's why I try not to drink caffeine at all, right? It's just cheating your wakefulness, but you're going to crash because it's just using the same sugar reserves. So stimulants increase blood flow to your brain and to your muscles so you can think and move faster, so to speak, or feel like you are, and they decrease blood flow to the extremities. That's why they can get cold when they do that. The opposite of that is, of course, alcohol. If we look at the alcohol, it's a depressant. It causes vasodilation. You can always tell when somebody is a lifelong alcoholic because they'll have a big red bulbous nose. That's because the alcohol, of course, causes the blood vessels in the outside, the periphery of their body to dilate, allowing more blood flow out there. That's why their skin feels warm, and it's why they make bad decisions. More blood is going to the outside and less to the vital organs like your brain. It actually causes vasoconstriction of your brain, which shuts off blood flow, and then you can't do things like walk the line backwards or say the alphabet backwards, and they always look at your nose and to see whether or not you are flush. It's a pretty good sign that you've been taking in something like a depressant. Now, this, of course, is a problem because then your brain doesn't function. You can't run a motor vehicle. And if you do it for your lifetime, it basically shuts down your brain in stages and you actually get less intelligent. You get dumber. Hey, now, normally at this point, I would demo the pig heart for you, which we talked about last time, and I'd get out some Dbags. And back to Dbag laying, this is where chapter 44 comes in. I would have a Dbag that was full of yellow dyed water and a bunch of salt water on the outside, super salty. And I'd have a Dbag with a bag that's just salt and water on the outside. In this one, the yellow water would actually push out of the Dbag because, well, there's more dye in, so it pushes from high to low, and the water would push out because water always follows salt. The bag would get smaller. In this one, the water would only push mainly into the bag until more of it reached in here, but it would push into the bag and the bag would get bigger because, well, water always follows solute, in this case, salt. This is a great explanation of how your excretory system works. It works solely by osmosis and diffusion, things pushing from high to low. So we'd be taking a look at GR44 at the end of class. We'd figure out the MVP and figure out that GR44 is all about this thing called osmoregulation, which you've already seen in your FRQ 2016, I think it was, right? This is about controlling our water. Osmo is the push of water. Regulation is keeping things balanced. And the main way we do that is simply just like Dbags. We don't really control it. It's controlled by concentration gradients. Water will always flow from high to low. And it's the water that tends to move because we have selectively permeable membranes just like the Dbag. And it will move from hypo to hyper, from more water to less water, from less salt, hypo, to more salt. It always follows salt. And that's crucial. That means that by changing your concentration gradients, you can change where water or anything that's in the water can flow. Notice the salt doesn't move easily because it's too big to fit through the holes. Now, things that we can think about this in terms of classes. When the bell rings, a lot of kids rush into my class because there's a lot of open space compared to all the people that are out in the hall. The desk would be like the solute. They're following that stuff. It's moving to a lower water potential, to something that's hypertonic. That, of course, can also work in your eye. Your eye will dry out because the air has very little water in it. That's a really low hypertonic solution. That means it's going to push out, and that's why water is constantly pushing out of your eyes, especially on dry winter days, right? Remember, the water always pushes from where there's more to where there's less. If you keep that in mind, it's good. From hypo to hyper. Now, that finishes off what we looked at this day. It's too bad you can't do the fish, but you can try it at home if your parents let you. If we look at the one thing that we're going to be going after is we're going to look at GR44 at the start of next week, and then we'll dig into the immune system, which is awesomely complex. The Fish Capillary Lab, look through the questions, make sure that you can answer those in terms of stimulants and relaxants and vasodilation and vasoconstriction. You should notice that your hemoglobin does some amazing things. It not only carries oxygen in your blood, it also carries CO2, protons, and aids in the plasma, the cytoplasm and the plasma, creating the stuff called bicarbonate ions, which act as a buffer to keep the pH of your blood from dropping too low and killing you. Partial pressure gradients control where things go. Hey, it's almost like everything pushes from high to low. This is easy. Okay, cool. Hope to see you at the AP review session. That's the end of this week's lessons. I'm tired. Hee hee hee.